Hello, scientists. It's time for another biology with Miss Judd. Last unit, we dove into the structure and the function of DNA, about how on the sides, it's our sugar and phosphate backbone that gives DNA its strength, and on the inside are those nitrogenous bases, and that's where the actual genetic information is coded. A's, T's, C's, and G's. The order of those bases means that I'm going to have brown hair, I'm going to have brown eyes, I can't see very well, so I need glasses. All of that is coded in our bases. And knowing the structure and the function of DNA, that DNA is the important genetic material, is super important because it does this amazing trick called DNA replication, and it's probably the most important thing that happens. If you think about it, each cell contains all of that DNA that we need to make other cells. We start out from a single cell and we end up with trillions of cells to make this body. During that process of cell division, remember mitosis, all of that information in the cell has to be copied, and it has to be copied perfectly. We don't want mutations. Mutations are bad news bears. And so DNA is a molecule that can be replicated to make the most perfect copies of itself, which is all the more amazing considering how many 3 billion base pairs of DNA have to be copied. Replication uses enzymes to make this a little bit faster. Today, we're going to dive into how DNA replicates itself so perfectly. At the end of this process, one DNA is all replicated and we have two of the exact same DNA. And then the cell can divide using mitosis, which is great. Sounds like we need to send it to Ghost Miss Judd. Hello, everybody. It's Ghost Miss Judd here to talk about our second lesson, DNA replication. Let's look at some of the big questions first of all. Who goes through DNA replication? That would be all living cells. Any cell which is run by DNA goes through DNA replication. This process is the process of copying all of a cell's DNA. This is the goal. This happens in interphase before a cell divides during mitosis. So if we are going to be copying DNA, that needs to happen in the nucleus of the cell. Remember that the nucleus is where DNA is kept safe. So if we are going to access DNA in order to make a copy of it, we need to be in the nucleus of the cell. The purpose is to pass on all the information needed to run a cell. There are 4 steps to DNA replication. The first step is to get to that genetic information, those bases. So we have to open up the double helix. Remember that it's hydrogen bonds that hold our bases together. So there's this player called DNA helicase. DNA helicase unzips the helix, essentially plowing through and opening up those hydrogen bonds. Once DNA is opened, another enzyme, DNA polymerase, adds new complementary nucleotides to each side. Remember those rules of base pairing. A goes with T, C goes with G. Let's go ahead and, with 1,000 stars around it, write that. The base pairing rule is that A goes with T, C goes with G. Polymerase adds in those complementary bases. This continues to happen up the strand of DNA, and then DNA polymerase proofreads and fixes errors. Remember that DNA replication needs to be an exact copy, and if something goes wrong, that's called a mutation, and that actually is really bad for cells. So polymerase comes back in and double checks to make sure that everything is right, that the A's went with T's, the G's went with C's. Finally, the 2 strands separate, which means we end up with 2 DNA molecules. So this is what it looks like as we go through. We have helicase opening up, polymerase adding in bases, and if I have on one side TACGTT, then my polymerase is going to add in an ATGCAA, that complementary base. This process is called semi-conservative replication. This means that each new daughter DNA molecule has one new strand and one old strand. Being semi-conservative, you save part of it. We don't want to just make an error by writing it down wrong. By adding in bases to an already existing side of the DNA molecule, we're more certain that this will be an actual exact duplicate. Now, I talked about helicase and I talked about polymerase. Those are enzymes. Enzymes are proteins with a very specific function. They act as a catalyst, meaning they speed up reactions that happen in cells. So, DNA helicase speeds up opening the DNA molecule. DNA polymerase quickly adds in the complementary bases. And we can see that happening here in this video. We have helicase running down the DNA and then polymerase coming in and adding in bases. And that's the basics of DNA replication. It's now time to send it back to regular Miss Judd. Thank you, Ghost Miss Judd. That was informative as always. Now, I know that was a lot of material, a lot of us to write down. What I need from you, though, is to just pause, look it over, and see if you can do that practice. The practice is an important part of this process. We can blindly write down what we hear and see, but until we apply it to something different, it's not going to stick in our brains, and we really want to remember this. So take the time, use your notes to work on this practice. You guys are awesome. Have a great day and make good choices.